Okay, so this is our video introduction to diffusion and osmosis. And a good place for us to start would be with a simple definition. It's a really basic idea. Now, a lot of students will read through a definition from the textbook and they'll take home the idea that stuff is moving from high to low. And that's okay, but I want to make that a little bit more specific because what's moving from an area of high concentration to low concentration is the solute. And this is an important distinction because in any given solution, we have both a solute as well as the solvent. Now, a solvent in this case, in the case of most biological systems, the solvent is going to be water. And as we'll find out later, if the solute is moving from an area of high concentration to an area of low concentration, at the same time what's going on is the solvent is moving in the opposite direction. If we have a high concentration of sugar on one side of the system and a low concentration of sugar on the other side, let's say this is happening in my swimming pool. There's no circulation. If I have a high concentration on the left side and a low concentration on the right side, what's going to happen is that the sugar particles will, over time, move in the direction from a higher concentration to lower concentration. Water molecules, on the other hand, remember that the solvent is water. You can sort of visualize all the bazillions of water molecules in the system. Water molecules actually are at a higher concentration on this side of the system. And at the same time that we're achieving an equilibrium with the sugar concentration moving from left to right, the water is basically moving in a direction from right to left. Basically, we have an opposite movement of solvent and solutes. And the movement of solvent is what we're going to be talking about as osmosis. Okay, so basically a couple of things that go along with this. The driving force for diffusion. The driving force for diffusion is kinetic energy. This is a really important concept for you because, what is it that makes the sugar molecules move from the left side of the swimming pool to the right side of the swimming pool? Well, it's actually the kinetic energy of the water molecules that are moving around, and also the kinetic energy of the sugar molecules. Water is moving around, and the molecules of water move. Their movement, their net energy, is what is transferred to the molecules of sugar. And so a sugar molecule, which is much bigger than a water molecule, is going to have its own movement. Some of these sugar molecules are going to be moving in one direction, the other direction. The faster these molecules move, the more net movement that we'll have from left to right in the case of the sugars, and right to left in the case of the solvent. Okay. Now, here's an important concept, because we actually have a pretty easy way of describing the net kinetic energy of an overall system. And the word that we have here is temperature. Yeah, the temperature of a system is basically a measurement of the average kinetic energy of the particles in the system. What that means is that if the swimming pool is very warm, that means we have a higher level of kinetic energy, which means that the particles are moving faster. We'll have larger movements. The particles are going to be moving at a much faster rate, and as a result, these sugar molecules are going to disperse at a faster rate. If the temperature of the pool is cold, then these particles are moving a whole lot less, and the speed in which these sugar molecules are going to disperse across the pool is going to be much lower. We probably saw some of this last week when we were looking at the effects of temperature on the activity of the enzyme. An enzyme, to a certain extent, is going to be assisted by warmer temperatures because the higher kinetic energy of the system is going to make it more likely that a substrate molecule will be encountered by an enzyme. Anyways, the bottom line here is that this is one of the key features that determines the speed at which diffusion takes place. Warm, higher energy, faster diffusion. Cold, lower energy, slower diffusion. The final state at which we don't have any more net diffusion is going to occur when the particles are uniformly distributed throughout the entire system. So if we scroll back up here, if you think about my swimming pool, this diffusion is going to be taking place until we end up with an equal distribution of sugar and water across the entire swimming pool. Once we have equal concentrations throughout, then we would say that's at diffusion equilibrium. That's where diffusion stops. You could say that when the particles of solvent and solute are uniformly distributed throughout the entire system, then we don't have a concentration gradient. We don't have high sugar in some parts and low sugar in the other parts, and at that point we don't have any further diffusion. So basically what we're dealing with in one of our experiments is going to be a system in which we set up a diffusion gradient, a concentration gradient. We're going to be putting a drop of blue dye in a beaker of water. It's going to be an area of very high concentration of blue dye in this one area of the beaker, and over time the particles are going to diffuse out further and further. And then you're going to be seeing the system reach equilibrium where you have an equal distribution of blue dye throughout the entire beaker. The time between which we actually have a concentration gradient where we add the drop of blue dye until we have diffusion equilibrium where the blue dye is uniformly distributed throughout. That's going to be the way in which we determine how fast diffusion is taking place. So that kind of covers our first little experiment in our laboratory today. Okay. And there's another part of the activity in which we'll be looking at another key feature that determines the speed with which diffusion takes place. Because if you think about all these water molecules have their kinetic energy. They're moving around at specific speeds, bumping into each other, bumping into the solute particles. Okay? And what's really moving in the case of diffusion are the particles of solute. And so if you visualize there being kind of a small solute, a molecule that's maybe not that much bigger than a water molecule, they're going to be moved relatively a lot. We'll have a considerable amount of movement on the part of these smaller particles. In contrast to what you might expect for a larger molecule, these larger molecules, their kinetic energy is determined by the water molecules that are bumping into them. But because they're so much larger, they're not going to be moving as much. They're not going to be achieving the same speed as the smaller molecules are because they're that much bigger. If you push a gigantic mass, it's not going to be moving nearly as much as a smaller mass would if you pushed it with the same force. Right? So there's another part of our experiment today in which we'll be setting up these little test tubes, and we'll have 3 test tubes, and we'll have like a really large solute diffusing across these test tubes are going to be filled initially with kind of like an agar medium. It's kind of like jelly. Right? So we have these tubes with agar, and we've got a little layer of water across the top. A little bit of water across the top. It's kind of like the stuff that you end up with anyways after you put the agar in the tube. A little bit of water will rise to the top. And then you'll be putting 3 different kinds of dyes on the top. You'll be putting the Papa Bear dye over here, you'll be putting the Mama Bear dye over here, and you'll be putting Baby Bear dye over here. One of them is going to be really big, one of them is going to be really small, and one of them is going to be a dye kind of in the middle range. And what you expect here is, well, pretty straightforward, right? One of these dyes is going to be moving faster, one of these dyes is going to be moving slower, and one of these dyes is going to be moving really, really slow. Or at least that's our expectation. And the distance that the dye travels in a fixed amount of time. So if Baby Bear dye travels that far, Mama Bear dye travels that far, Papa Bear dye travels that far, the distance that they travel, it's going to be our metric of the diffusion speed. Okay, so we're going to be graphing out a plot. We'll be saying, well, how fast does a dye particle move? It's going to be depending on the size of the molecule. So I guess your question here is going to be, what is going to be the independent variable? And which of these measurements is going to be the dependent variable? So, okay, go ahead and tell me, what is the thing that I put across the bottom? What am I going to be putting across the bottom of my graph? And what is going to be the thing that I put on the vertical axis? What's going to be measured and graphed on the vertical axis over here? And what's going to be graphed on the horizontal axis? Okay, and in another part of our experiment, this part is the one that's kind of fun. It's going to involve another threesome. It's going to like Mama Bear, Papa Bear, and Baby Bear again, only this time we'll be talking about cubes. We've got agar cubes, that same jello-like material. We'll have a big cube, a medium-sized cube, and a little cube. Mama cube is here, and Baby cube is going to be here. Okay, so these are 3 cubes, and in the agar we have a substance called phenolphthalein. Phenol, and here's the fun part. It's a phthalene, but it's got a silent pH. Phenolphthalein is another one of those color indicators that turns bright pink or fuchsia when it gets into a high pH, but when it's at a neutral pH, it's clear. Okay, so what we have here are these tubes. They're at a normal pH initially, and then we're going to be dipping these tubes into sodium hydroxide, NaOH. And NaOH has got what kind of pH? Yeah, of course, it's basic. When these cubes hit the NaOH particles are going to start diffusing inwards. They diffuse inwards, inwards, inwards. Basically, they're moving through the agar in the same way that the dyes are moving through the agar in our dye experiment, right? And what's going to happen is that depending on how large the cube is, the NaOH is going to be diffusing in either to entirely fill the cube, to partially fill the cube. Basically, what's going to happen here, which you should note here, I guess the bottom line is that the smaller cube is not basically taking in the NaOH at a faster rate, but because all of its parts, because even the center bit of this cube is relatively close to the outside edge, it's able to completely turn the color that's going to be associated with the NaOH. Okay? Whereas these larger cubes, they're going to have the NaOH diffusing into the center more or less at the same speed, the same diffusion speed, but there's going to be an area in the middle that doesn't actually get the change in pH. And the bigger the cube, the bigger the area inside the cube that's not going to be having been affected by the change in pH. Okay? The bottom line here is kind of a simple one, which is that cells take up nutrients kind of in the same way that these cubes are taking up NaOH. It's by simple diffusion across the outside surface. So if you have a really large cell, the food might be diffusing in, but it's not really getting to the center part of the cell. There's a large area in the middle of the cell that is effectively starving to death. So our lesson here is basically that cells are small. One of the reasons why cells are small is because they rely on diffusion from the exterior surfaces to get their material, to get their food, for all the food molecules that they take up to distribute throughout the entire cell. If cells grew in size, then they would lose that surface area. We'd end up with a big void in the middle of cells that would not be getting any food. So instead of having a cell grow double in size and triple in size or triple in linear dimension, what we typically do is have one cell that will grow and it'll divide and it'll end up with 4 cells or 8 cells, each one of which has the same surface area as the one that we started off with. Right? So in other words, if we were to increase our volume but also increase the number of cells, we're not sacrificing any surface area relative to the overall amount of volume. Okay? So this is basically our first installment of surface area to volume ratio. We'll be emphasizing this again in class. The last of our experiments, and we kind of have to do these experiments simultaneously because of the nature of diffusion. It takes a while for diffusion to occur, and so you have to set up an experiment and then allow for some time for diffusion to occur and then come back to it later. So we're not going to be doing the first one, waiting till it's done, and then starting the second one, waiting till it's done, doing the third one. If we did that, it would take more than the 3 hours that we have. So we're going to be doing a little bit of overlapping of these experiments. We'll be having to start one experiment, start another one, come back to the first one, come back to the second one, then maybe start the second one, the third one, and the fourth one similarly. The last experiment is really kind of neat. So in this experiment, we'll be creating an experimental system in which we have an artificial cell. Okay? And the artificial cell is going to be composed of dialysis membrane that's going to be replacing the membrane of the cell. And dialysis membrane is the same stuff they use in dialysis treatments. That's the treatment that people undergo if they have a malfunctioning kidney. We're able to remove a lot of the impurities and toxins from the blood via an artificial kidney where the blood is moved through this dialysis machine and it returns back to the patient in a much cleaner state. So our dialysis membrane is going to take the place of the cell. And what a dialysis membrane is it's a hollow tube. It comes in as a flat place. But it's a hollow tube, and what we need to do is we need to create an artificial cell. So we're going to be putting a little clip here, cutting it off, filling the dialysis membrane with a solution, putting a clip on the other side, closing it off. And I've been asked by management to remind everybody, please do not throw away these orange clips. These clips are hard to find, and so we'll be using these clips to create a little sausage of dialysis membrane. It's going to contain an initial solution that has protein, it's got monosaccharide, and it has starch. Okay. That's the dialysis solution. We'll be closing off this one side, filling the bag with dialysis solution, closing off the other side, and then we're going to rinse it really well. We'll be using the distilled water squirt bottle to rinse this whole thing off really well so we don't have any of the dialysis solution on the outside. And then we're going to soak this whole thing in Lugol's iodine. And then what do we expect to happen here? We've got Lugol's iodine out here. And maybe the Lugol's iodine is going to diffuse inside. Okay? Maybe it's not. I guess the question is, well, will Lugol's iodine diffuse from the outside of the membrane to the inside of the membrane? We talk about the protein that's inside the dialysis solution, the monosaccharide, and the starch. We can ask questions on all four of these substances: protein, monosaccharide, starch. Does it diffuse? And we'll need to run tests. We'll basically need to run tests similar to the ones we did for the biochemistry laboratory in order to ascertain the answers to these questions. Okay. We're also going to have to run a control. The control is going to, we need to be able to validate each one of these conclusions. Basically, I want you to think about what kinds of tests we're going to run in order to determine whether or not Lugol's iodine or the protein or monosaccharides or starch, whether they're going to be present and diffusing into the portions of the system where we don't have them initially. The control that we do in order to validate this is going to be done as a demo. I'll probably be running a single control at the front of the class, and you'll be using the results from my control in order to validate the results that you have in your system here. So we'll be setting up one of these, and I want you to think about, well, how are we going to be asking these questions, and what kinds of controls are we going to be using to validate our answers that are coming from the first set of experiments. Okay, so I hope this helps you out. Make sure to come to class prepared, wearing appropriate footwear. We'll be covering a few of the safety issues in class, as I usually do, just in case there are some students that don't have a chance to view this video. Okay, hope you enjoyed it. See you in class.